Dear students, this video is talking about the gene expression regulation in prokaryotes and eukaryotes. As you know, gene regulation is how cells control the genes are turned on or off. It's like a switch that tells the cell which instructions to follow. This process is very important because it helps the cell save energy and resources, making sure it only makes what it needs. Let's start. In prokaryotes, like bacteria, gene regulation is often simple. These cells are small and do not have a nucleus. So, their genes are usually turned on or off altogether, depending on the environment. But in eukaryotes, like human cells, gene regulation is more complex. These cells have a nucleus, as we see here, where their genes are kept, and they need to carefully control gene activity. Each cell in a person's body has the same genes, but different cells might turn on different genes. Let's talk about prokaryote gene expression regulation. So we need to explain what is operon, which is a DNA unit in bacteria like E. coli that controls the expression of multiple related genes together, which includes promoter, where RNA polymerase binds to start a transcription. Second, operator. It's a regulatory site where proteins like repressor bind to control transcription. And repressor came from regulatory gene. After expressing the regulatory gene before the promoter, it will give the repressor protein. And third, structural genes, which encode proteins for shared function. We have two types of operons. We have lac operon and trip operon, the inducible and repressible. Lac operon. The lac operon is a genetic mechanism in bacteria like E. coli that regulates breakdown of lactose. Without lactose, the repressor protein, which is produced by the regulatory gene, it binds to the operator region, which blocks RNA polymerase from transcribing the operon's structural genes. And now, with the presence of lactose or the isomer allolactose, it binds to the repressor protein, and that will lead to conformational change in the shape of repressor, that preventing it from binding to the operator, and that will allow the RNA polymerase to synthesize messenger RNA to do the transcription. So, we will understand that the presence of lactose will induce transcription process. We call lac operon is inducible process. What about the trip operon? The trip operon in bacteria like E. coli regulates the production of enzymes involved in synthesizing amino acid tryptophan. With high level, tryptophan acts as a co-repressor and binds to the repressor protein. This activates the repressor, allowing it to bind to the operator, which blocks RNA polymerase. So the transcription is repressed now, stopping tryptophan production. But with a low level of tryptophan, the repressor protein is inactive and cannot bind to the operator. That means the RNA polymerase binds to the promoter and transcribes the operon structural genes. These genes code for enzymes that synthesize tryptophan. So we can say that the trip operon is repressible, not like the lac operon, which is inducible. Moving to eukaryote gene expression regulation, which controls how genes are turned on or off to make sure cells work properly and respond to changes. Let me explain this figure, which represents part of the mechanism. First of all, we can see the RNA polymerase already binds to the promoter as usual. But number 3 represents the activators that will build a complex and bind to transcription factors that include enhancers, regulatory proteins. Now, mainly, we have 2 sets of transcription factors. One set, as we see here, form complex that guide and stabilize the binding of RNA polymerase to a promoter to do the transcription procedure. The other set includes regulatory proteins that help controlling the rate of transcription. Actually, this process ensures precise regulation influenced by activators, enhancers, and transcription factors to meet the cell-specific needs. That's it. Thank you.